Quantitative Buffy coat test, simply QBC test. So this test is used to diagnose malaria, filariasis, and visceral leishmaniasis. So quantitative Buffy coat tests we are going to discuss in this video. QBC test for the diagnosis of malaria, filariasis, and visceral leishmaniasis. So this is the life cycle of malaria parasites of Plasmodium species, including Plasmodium vivax, Plasmodium ovale, and Plasmodium malariae, and Plasmodium falciparum. So these are the cycle. So there is a erythrocytic cycle and exoerythrotic cycle, that means liver cycle. Then there is a sporogenic cycle. So once the human is infected, it is enter into the liver cells, there is a liver cycle. In that case, there is a scissions will get ruptured, then it is enter into the blood circulation. Once you enter into the blood circulation, there will be a ring stage, that is immature trophocyte cycle. So this is having some diagnostic importance. Then it is converted into mature trophocytes and also gametocytes. So these both are having some diagnostic importance. Then it is converted into scissions. Then these scissions getting ruptured, so it is converted into the different malarial parasites, malarial falciparum, vivax, ovale, and malariae gametocytes. So again it is enter into the sporogenic cycle. There is a oocyte formation at the end of the sporogenic cycle. Then the mosquito release. Then again infecting the human by biting. So this is the cycle of malarial parasites. The diagnostic importance is there at the immature trophocytes and mature trophocytes and scissions and gametocytes. So these are the forms we can able to visualize through the microscopy. So what is the principle involved in the QBC test? So QBC test is based on acridine orange stain. So you know one thing, all the stain or dye having the property of binding with any material. For example, if any dye you mix or pour in the cloth, it will attach permanently with the cloth. So the same thing is happening in the QBC test. We are mixing the acridine orange stain or dye with the malarial parasite containing blood samples. So this acridine orange will bind with the malarial parasites, nucleic acid and cytoplasm. It will show the color difference. So in this case, acridine orange stain will mix with the blood samples. Then this blood sample is centrifuged. So centrifuge is the separation technique which separates the blood components according to their density. So after centrifuge, it is examined under the UV light fluorescence microscope. So this case, acridine orange will bind with the DNA and RNA. It will give the yellowish green fluorescence, and another way it will bind with the cytoplasm. It will give bright red fluorescence when excited by the blue light at 460 nanometer. So we can able to detect through fluorescence microscopy only, since the acridine orange will give the fluorescence light. It will emit the fluorescence light. So the basic principle involved in the QBC test is acridine orange bind with the nucleic acids and cytoplasm of malarial parasites. Once it is bind, it will give the fluorescence light. Yellow is green from nucleus, bright red fluorescence from cytoplasm. The other cells present in the blood, such as red blood cells, does not absorb the stain or dye, so it do not give any light or fluorescence. So that will make the difference between the malarial parasites and the other blood components present in the blood. So this can be visualized through the fluorescence microscopy. So this is the malarial QBC tube. So this tube is 75 millimeter in length. It's internally coated with potassium oxalate, acridine orange, and anticoagulants. This anticoagulants will prevent the blood clotting, which maintain the blood fluid nature. So there is another thing, it contains a precision molded float within the blood filled tube. So there will be a float, plastic float. So it will expand the content present in the inside the QBC tube. It will expand or enlarge tenfold. So you can see the enlarged portion of the blood components. So once this tube is centrifuged, the blood components will be separated according to the density, right? The dense material like RBC will be there in the down part. Then other cellular cells like platelets, lymphocytes, monocytes, granulocytes will be there in the middle. Then plasma fluid in the upper layer of the QBC tube. So the malarial parasites usually present in this cellular component, less cellular component, platelets, lymphocytes, monocytes, and granulocytes. So procedure involved in the QBC test is just drawing the blood samples in the QBC tube by capillary action. Just we need to keep the capillary tube in the fingertips, so this absorb or it will uptake the blood into the tube by capillary action. Then rotate the tubes for 10 seconds to dissolve the contained residues in the blood. Then inserting the cylindrical insert or plastic float inside the acridine orange coated capillary tube. So then we may insert the small plastic float inside the tube. Then we may go for centrifuge at 12,000 revolution per minute for 5 minutes. So after centrifugation, we may able to observe in the fluorescence microscopy. So you can see the finger, the capillary tube kept at the finger, it is oozing the blood. Then this tube is ordered with the float, plastic float. Then we go for centrifuge. So you can see the small tube is enlarged. This float is enlarging the particular point where the cell contents are there. So it is enlarged view of the float. So results and observation. So in this case, the results, this is the enlarged view of the float. It's showing the blood components. So first of all, there will be a crescent-shaped gametocytes. So you can see the gametocytes present in the platelet layer, platelet or lymphocyte or monocyte layer. There will be a gametocytes present in the upper layer of the float. So the gametocytes, the first one, it will appear near the interface of lymphocytes or monocytes and platelet layer. The second is the senescence and mature trophocytes. You can see the senescence and mature trophocytes. Then another is the ring form, ring-like mature trophocytes, immature trophocytes, ring-like structure of immature trophocytes. There is a ring-shaped immature trophocytes will appear throughout the red blood cell layer, RBC layer, right? You can see the RBC layer, there will be a ring forms or immature trophocytes present in the red cell layer, down part. This is the somewhat a ring shape, it will be there in the down part. So according to the density, the cell components are present there. This float is enlarging the blood components. So by the time we can able to see the malarial parasites through the fluorescence microscopy with the help of the dye acridine orange. So the acridine orange nucleic acids are give yellowish green color fluorescence. Then cytoplasm of the malarial parasites will give the red fluorescence. Thank you.